All right, this next section of video will be about the cytoskeleton. This does get a little complex, and I definitely will say there's a lot of information on here that is probably beyond the scope of the exam, but I still want to give you an overview of it. Okay. Just like your skeleton, a cytoskeleton has very similar jobs and very similar, somewhat similar setup. You don't really see it a lot in these basic textbook drawings of cells, but there are thin sort of lines of cytoskeleton inside the cytoplasm of the cell. So you don't really see it too much in most textbook pictures, okay? So it's one that's easily missed. When we take an electron microscope picture of this is a quarter of a micrometer. So we are really zoomed in here. And these are so thin that light microscopes don't resolve this stuff very well because it's so small, so thin, okay? There are some parts that are thicker than others, but it's a big mesh of proteins. What's it do? Just like a real skeleton. So remember, functions are what we're really after. Structure and shape are sort of the big ideas, but it has some other more subtle functions we're going to get into. Okay? They allow cells to move. A lot of cells require movement, and they give anchorage points, and also they actually burn ATP to move. It also allows things to move around in cells on little sort of railroad tracks, okay? And we'll talk about that. When cells divide, okay, so let's say we are undergoing mitosis up here. They help the cell separate organelles apart, especially the chromosomes, okay? Especially chromosomes. Is that too fast? Well, all right, so movement, division, structure, and shape. Those are your big ideas. Movement, structure and shape, and division and cell walls. Okay. What are these things made of? When we look at them up close, they come in a couple different varieties. And the first one is microtubules. The second one is microfilaments. Don't write these down because I'm going to go over them individually. It's okay if you wrote the first one down, but we're going to kind of look at them individually. This is just where we're going. So let's look at these three different types of cytoskeleton. Okay, three different types. The first one, microtubules. These are basically spirals with hollow space in the middle, and that's why they're called tubes, because they're like a straw or a tube. They're hollow in the middle. These help cells keep their shape, and they also, we'll talk about, when we talk about cell walls, these will come up again. Microtubules help cellulose fibers align themselves. Okay, these are tracks, like I said, railroad tracks, okay? And they help what are called motor molecules move things around in the cell, okay? So if you want to move something from one part of the cell to another, one of the ways that can happen is on these motor tracks. I'm going to turn my light back. Oh, it's not that bad. So let's talk about the railroad tracks. All right, so you'll have a tube, which is shown right here. And you have this little walking protein that kind of walks like this, okay? It will walk this way, and then it'll walk this way. It kind of does this sort of twist movement. I'm going to put a video of this on my site, but every time that happens, every time it makes that step, it burns an ATP. And the cargo, what it's moving, is contained in a vesicle up here, and it will literally drag this along this track right here. And so here's your electron microscope picture of it. These are vesicles. It doesn't really show the motor protein that does the walking. It's best shown by an animation I'll post later. Okay? So those are like railroad tracks that move cargo around the cell. So that's one of the functions of microtubules. What else do? They make cilia and flagella, okay? Cilia are little hairs, and flagella is like a whip. Let's take a look at that. They also help things move inside of cells. Now, this is a little different than the railroad track idea, the microtubules with the motor proteins. They also make up what are called centrioles. And if you remember, have you guys done mitosis in sophomore bio? Centrioles are little sort of shapes that spindle fibers connect to when they pull chromatids apart in anaphase. So these are centrioles right here. Okay, and when we get into mitosis, we'll talk more about that. But they also make up the other parts of that. They make up the spindle fibers. So these things, these yellow guys right here, they're also part of that. All right, let's talk about what cilia and flagella are. Okay, cilia are short, but they're numerous, and a lot of times you'll see single cells covered in little tiny hairs, and they're almost like little oars on a rowboat or a big Viking ship or something. And these will all beat in coordination, and a single cell can swim with these really well. Now, they're also found on animal cells too, but they're not for locomotion. Okay, if this was your trachea is lined with little hairs, and they are cilia. And their job is to move mucus and dust and whatever you get into your respiratory tract. Their job is to move that stuff up and out. Okay. A lot of times, you'll hear people get out of bed in the morning. They're like, and what they're doing is while they were laying down, that whole thing was sideways and no gravity. So you have a lot of gunky mucus at the top of your throat in the morning that a lot of people will cough up and spit out. Now, here's something interesting. If you are a smoker, okay, smoking kills these. Okay, smoking kills these. And so when smokers get respiratory diseases like colds or bronchitis or pneumonia, they tend to be sicker much, much longer because they don't have any cilia to move that gunk out. Okay, so that's something we don't want to do is smoke, obviously. All right, flagella means whip, and that's going to be more like a long tail on a cell. Now, there are protists that use these, but most commonly we think of sperm cells that use those. These are basically, like I said, to help them swim or to move things around inside of an organism. Okay, here are some pictures of this. You've got these big tails. Sperm aren't the only cells that have these, and this is the only human cell with a flagella. Okay, now like I said, your respiratory tract has cilia, but here's a little protist here with all these little hairs to help it swim around. Okay, how do they do this? They have a little segment in them that's similar to like muscle tissue. It's called a motor protein. Now, you don't have to write that down. I don't really care that you understand how these are put together. The 9 plus 2. Let me show you what that means, okay? They do have this little protein that's really interesting and I want you to kind of pay attention to. It's called dynin, and a dyne is a unit of force before we had the metric system. It's a very small unit of force, okay? No, I'm thinking of an erg. Well, I can't remember. I'll have to look those up. An erg and a dyne. Those were really pre-metric system units of force. All right, how do these work? You have 9 pairs of tubes, okay? And they're pairs of 2. And they're wrapped around 2 tubes in the middle. Now, that's nice and pretty. It kind of looks like some kind of fancy pasta. But between them, you have these little proteins that when they get hit with an ATP, they squeeze. Now, they don't always squeeze at the exact same time, so that can lead to like very sort of complex movements going through these fibers, okay? Now, this is way above my education as far as like understanding how the order of these is programmed to move, okay? But I do know that it happens, and that happening in a coordinated way allows these flagella or these cilia to beat in a very regular way, which allows microorganisms or cells to either swim or move stuff. Okay, here's sort of a picture of that happening. And that little dynin protein hits by an ATP, squeezes, and bends the microtubule. All right, centrioles, we talked about those. There's usually one pair per cell located close to the nucleus, except during cellular division. Okay, these are mainly an animal cell thing. So if you want to write that down, one of the big things that seems to show up quite a bit in this class is the difference between animal, plant, and prokaryotes. All right, I don't care that you know the structure, 9 sets of triples, okay? But I do want you to know this, okay? Cell division. I want you to know that they are cell division. Now, they are 9 sets of triples, so they'll be arranged in a set of 9 triples. Okay. We don't normally see these in cells, so I want to show you this picture of stained microfilaments. So they do show up throughout the whole cell, okay? They're all over the place. You just normally don't see them. All right, what all kinds of things do these do? These allow muscle contraction to happen with filaments called actin and myosin. These allow what's called cytoplasmic streaming, okay? And just like a stream is like water that flows, if you have a cell, you might want the cytoplasm to circulate, okay? And that would be to move things around the cell, to get things mixed up in the cell. Plant cells do this around their vacuoles. They also help certain protists called amoebas. That's one example. Okay, so this is a cell that can kind of blob all over the place. When it wants to send out what's called a pseudopod, it will push cytoplasm to one side, and then it can sort of blob over something it wants to eat. So it can kind of sneak around here and engulf that. All right. Cleavage furrow formation. Cleavage just means crack, okay? And so when cells start to divide, they'll start sort of pinching together right here, and that happens due to these this part of the cytoskeleton. Anything that has to do with changes in cell shape is going to be part of this. So these functions: muscle, streaming, pseudopods, cleavage, and anything that changes cell shape, which is what all these up here do, okay? These are all changes in cell shape. Okay, so here's some pictures of examples, okay? We are pushing cytoplasm to one side. Here in a plant cell, we're circulating around the vacuole. All right, intermediate filaments. These are sort of the middle-sized filaments. And they're made of proteins that are in thicker cables, okay? These aren't tubes. They're thicker. And they kind of have a ropey appearance. I don't know. It kind of looks like a big rope. Almost braided, but not quite. Now, these are shown in this picture right here, and you can kind of see these are microfilaments, which are the smallest. These are microtubules, which are hollow in the middle. And intermediate filaments are shown here with that rope-like structure. And let's go over their shapes, okay? Microtubules, mainly moving things around, okay? It does do some cell shape, but these are mainly movement, these microtubules. Microfilaments, also movement. So these two have sort of the same structure. These ones here are more about maintaining structure, okay, shape. So that's just an overall summary of the different types of cytoskeleton. All right. Just an overview, okay? It changes a lot. That's what dynamic means. And I just want you to know, a cell is just not a bag of liquid. A cell is going to have lots of these fibers in it to give it structure and to help things move. Structure and movement on a cellular level.